Okay, well, as we get started this evening, I want to have a little bit of fun. No sense in having an hour-long lecture of boring science stuff, so let's have a little bit of fun. And I'm just curious this evening, how many of you have seen the Jurassic Park or the Jurassic World movies? Quite a few of you have, right? Well, I saw one person shaking their head no. Well, we're kindred spirits because I've never seen any of the movies. And when people find out that I've never seen Jurassic Park or any of the Jurassic Park movies or the Jurassic World movies, they are shocked by that because they think that because I'm a biochemist, I would want to watch those movies, right? Because it's all about cloning dinosaurs from ancient DNA, and cloning and DNA is kind of biochemistry-like. And so they figured as a biochemist, I would want to watch those movies. Well, I have really no interest in them whatsoever. And it's not because I think that the movies aren't communicating an important message. They are in a very entertaining way, to be certain. And that message is really, what are the limits of science, right? Those are very, particularly in this day and age, where we have emerging biotechnologies, those are very important questions to ask. But to me, when I see these movies, I just see them as monster movies where you have dinosaurs terrorizing people. And I'm not really all that interested in watching dinosaurs chomp on people. Now, if you like that, that's okay. I'm not judging you. Have at it. Enjoy the movies. But it's just not my cup of tea. Now, lest you get the wrong impression, one of my favorite TV shows is The Walking Dead. So I'm okay watching zombies chomp on people, but I have no interest in seeing dinosaurs do that. But anyway, a couple of years ago when the latest Jurassic World movie was released, our publicist, Anastasia Young, and maybe some of you have interacted with Anastasia over the years, approached me and said, with the Jurassic World movie coming out, if you're willing to do interviews on the movie, I could get you some media appearances, and would you be willing to talk about scientific errors in the movie? And I thought, well, sure, I'm game. But then I thought, oh no, it means I'm going to have to watch the movie. And then I thought, I know what I'll do. I'll just Google it. And so I just Googled scientific errors in Jurassic World. And so there's a number of articles written by paleontologists who are complaining about all the scientific mistakes in the movie. And so just for fun, and hopefully not to ruin the movie for you, I thought we could just look at a few of those mistakes. And one of them has to do with the way that dinosaurs are depicted in the movies. Dinosaurs, we now know, had feathers. And in fact, paleontologists will categorize dinosaurs as being both avian and non-avian. And both groups had feathers. In fact, paleontologists who are working from an evolutionary framework would argue that the avian dinosaurs gave rise to birds. In fact, they would argue that dinosaurs really didn't go extinct. They actually are still here with us today. We just call them birds. And so they see this evolutionary connection. But both groups had feathers. Now, the director and the producer of Jurassic World decided against feathering their dinosaurs because it would disrupt the continuity with earlier movies. And to be fair, when the earlier movies were made, we didn't know that dinosaurs were feathered. So this is a recent discovery. Another error was the way the dinosaurs sound in the movie. They roar, but in fact, paleontologists think that dinosaurs actually sounded like birds, not like mammals that would roar. The dinosaurs in Jurassic World are depicted much larger than they actually would have been in real life. And of course, this makes sense because you're making a monster movie, so you want the dinosaurs to be ferocious. Now, here's another error that's amusing to me. And again, I've not seen the movie, so I'm only reading the accounts of this scene. But apparently there's a scene in the movie where people are trying to get away from a T-Rex, and they have this idea that if they stand perfectly still, you can see where this is going, right? Why this is an error. If they stand perfectly still, the T-Rex wouldn't be able to see them because T-Rex can only pick up movement, right? And well, that's not true. The T-Rex would have seen them whether they were moving or standing still, just like we can see things that are standing still or moving, right? And so just as a public service announcement, if you're ever being chased by a T-Rex, keep running. You don't stand still. That's not going to do you any good, and it's just going to make the dinosaurs work that much easier. Now, the last error that I want to talk about real quickly is the queen mother of all errors, and that is you can't clone a dinosaur even if you had ancient dinosaur DNA. Now, I'm not going to get into the technical reasons why you can't do that. If you're interested in the Q&A, we could talk a little bit about that. But the entire premise of this movie franchise is built upon a pretty significant scientific error. Now, this may be disappointing to some of you who were hoping that maybe one day you could actually see a real dinosaur that would have been cloned from ancient dinosaur DNA. And I don't think that's going to happen. But it doesn't mean that you can't maybe one day interact with a dinosaur, because there's actually a serious scientific proposal on the table to actually try to resurrect dinosaurs from extinction. And the idea here, again, is connected to what I mentioned earlier about the presumed evolutionary connection between birds and dinosaurs, where there are scientists who are thinking that through techniques in molecular biology and developmental biology, we could actually reverse the evolutionary process and revert birds into dinosaur-like creatures. And the bird that has been selected as the model for this research program is the chicken. And so there are scientists that are trying to create something called the Chickenosaurus. You laugh, but this is actually serious. This is a serious scientific proposal. Now, if you think about it for a minute, this makes a lot of sense because of the poultry industry. Because of the poultry industry, we understand chicken biology inside and out. We have the chicken genome that has been sequenced. We know how chickens grow and develop. And so this is valuable information that could be leveraged now to reverse evolve chickens into into a dinosaur-like creature. In fact, one of the scientists who is championing this idea is Jack Horner, who's a famous dinosaur paleontologist at the University of Montana. And as an interesting aside, he was the real-life scientist that was the inspiration for the Alan Grant character in the very first Jurassic Park movie. But he wrote a book a few years ago I highly recommend the book. It's just absolutely an entertaining read, called How to Build a Dinosaur. And I love the subtitle, Extinction Doesn't Have to Be Forever. That's a great subtitle. But Horner is essentially one of the scientists that's championing this idea. And here is a cartoon showing essentially the difference between a chicken and a perceived Chickenosaurus that people are trying to build in a laboratory setting. And this is essentially a slide showing the strategy where the researchers are looking to manipulate the developmental process so they can modify the development so that a chicken will develop a tail, will develop legs that are like a dinosaur, feet like a dinosaur, forelimbs like a dinosaur, and then a skull in the shape of a dinosaur. And again, if you think that this is kind of absurd, there's actually scientists that are working diligently on this problem. For example, a team of researchers from Harvard University a few years ago did a really interesting experiment with chick embryos where they had little microscopic beads that they coated with growth factors, and they could implant them in the regions of the skull during the course of embryonic development. And if they implanted them in the right locations at the right time in development, they could actually keep the beak-like structure of the chick from forming, and instead it would form a skull that was much more like a reptile. And so this is the chick skull, this is a reptilian skull, and this is what they were able to produce, again, with these manipulating the developmental process. Not all the way there, but they made significant strides demonstrating in principle this kind of manipulation is possible. A research team from Chile published 2 papers where they used a similar kind of approach to modify the foot structure of chickens to be more like that of dinosaurs. And they also modified the leg structure of chickens to be like a dinosaur type of leg. And one of the big differences between birds and dinosaurs has to do with the size of the fibula. You know, in the lower bone of vertebrates, in the lower leg, there are two bones. There's the tibia and the fibula. And the fibula in birds is like a needle-like structure where the bone no longer makes contact with the ankle bone. And so they were able to essentially modify the development of the chicken leg so it would look like that of a dinosaur. So this idea is actually not that outlandish. And it could very well be that in the near future we could actually have dinosaur-like chickens around. We could have chickenosauruses around. And so if you're ever walking down the streets of Los Altos and you see a chickenosaurus stomping around, my advice is run, don't stand still, hoping that it won't see you, right? Now, we've played around for 15 minutes here, having some fun about this work. But this type of research is actually part of a very exciting area of biology today called synthetic biology. This is one of the hottest areas in science today, and this area involves the interface of engineering with biology. It's kind of like an approach to science that fuses, again, engineering and biology. And the goal in synthetic biology is to create artificial, non-natural life forms. And there are two approaches that are employed. One involves re-engineering existing organisms. So trying to convert a chicken into a dinosaur-like creature involves re-engineering that organism. And so that's one approach that can be taken. And so people are not only trying to re-engineer animals and plants, but also a lot of the work is involving re-engineering microorganisms, bacteria and organisms like yeast. There's also another approach that involves essentially chemistry, where scientists are trying to create protocells, starting with simple chemical materials, and trying to complexify them and get them to aggregate into protocellular entities that assume properties of life. And so this is called the bottom-up approach. But this is, again, a very exciting and active area of work in biology today. And the idea behind synthetic biology is really important because many people see this as being the next wave in technology, that this is going to be driving another revolution in technology where we could create organisms that would, in a sense, be like bioreactors, where you could, in this case, in this diagram, take a bacterium that you've re-engineered or have modified so that bacteria could take simple raw materials from the environment and then convert them into highly valuable products like drugs or biofuels or bioplastics. Some people have the idea that you could maybe engineer microorganisms and use them for agricultural purposes where they might form symbiotic relationships with plants, helping those plants to be more productive, or that they would help to control pests in the environment. Some people think that you could design microorganisms to remediate pollution in the environment. So this is going to be potentially the next revolution in technology. But there's another reason why researchers want to do work in synthetic biology, and it traces back to the statement made by Richard Feynman, the Nobel laureate in physics. And I'm going to paraphrase what Feynman said, but he said something to the effect that if I can create it, that means I understand it. And so the idea here is that if we can create artificial cells in the lab, or if we can re-engineer novel organisms, new life forms, it means that we basically understand life that much better. It's going to lead to a deeper understanding of biochemistry and biology. But there's also people that look to synthetic biology as a tool to help understand the question of origins. So for example, with the bottom-up approach to synthetic biology, where people are trying to create protocells, well, what are they doing? They're starting with simple molecules, and they're going in the lab and they're trying to get those molecules to assemble into protocellular entities that assume the properties of life. And this typically involves stepwise reactions where you're, again, complexifying the system and getting things to aggregate. Well, scientists that are trying to understand the origin of life, where we see chemicals on the early Earth presumably evolving into the very first cells, are doing essentially the same thing. They're trying to recapitulate the different steps in the origin of life, and to do that involves, again, going in the lab and trying to create greater complexity from simple molecules and getting those systems to aggregate. And so origin of life work from the bottom up and synthetic biology from the bottom up are in effect the same activities. And today, a lot of the most interesting work in origin of life research is actually synthetic biology. And many origin of life researchers that are working on origin of life problems are also involved in work in synthetic biology as well, where these two areas are now intertwined and interplay with one another. And the idea is that if we can understand how to create life in the lab, this is going to provide us with insight as to how potentially life could have evolved. People are also looking to synthetic biology to understand the history of life on Earth. How did life evolve on Earth? So the experiments on the Chickenosaurus are essentially an example of synthetic biology used to address questions in evolution, where this idea of reverse evolution is now becoming a popular experimental strategy to understand the evolutionary process, where suddenly evolutionary biology has become a laboratory science. Now, when you talk about doing this kind of work, where we're creating artificial, non-natural life forms, there's all kinds of philosophical implications to this work, right? And the immediate one, probably in your minds, is should we play God? Right? That's the first question that everybody asks, which is essentially the question that the Jurassic Park and Jurassic World movies were asking. Well, I'm not going to address that question tonight. We could maybe talk about it in the Q&A. But there's another philosophical implication that I am going to focus on this evening, and it has to do with our place in the cosmos. How do we see ourselves in terms of our place in the cosmos? And a number of years ago, in an interview for the Associated Press, a philosopher by the name of Mark Badeau, who at that time was the chief operating officer for a company called Protolife, which was headquartered in Venice, Italy. I assume it's still there as a company, and that Badeau is still part of that operation. But he was interviewed because this company was looking to make protocells. And he's a philosopher by training, and this is what he said. Creating protocells has the potential to shed new light on our place in the universe. This will remove one of the fundamental mysteries about creation in the universe and our role. We are doing things which were thought to be the province in some quarters of God, like making new forms of life. And there are many origin of life researchers that I interact with who actually will echo this kind of sentiment, to the effect that if we can create life in a lab, then it means there's nothing fundamentally special about life. And that if we can make life in the lab, it means that it's all that much more reasonable that life could have evolved on the early Earth through chemical evolution. And so many people see success in synthetic biology, particularly the bottom-up approach to the origin of life, as being, again, validation of the evolutionary explanation for life's origin. Now, to me, I think there's a real irony here, and you probably have picked that up. And that irony is this, that the work that's being done, for example, with the bottom-up approach to the origin of life, actually does shed an understanding of how life emerged on Earth, but I think it actually undermines an evolutionary explanation and instead indicates that there must be a creator that's responsible for the origin and the basic design of life. And the reason why I would say that is that when you look at the work that's done in synthetic biology to try to create protocells, what becomes evident is that you have some of the best minds in the world developing these very sophisticated, elaborate strategies that are based on hundreds of years of scientific knowledge that rely on foresight and then require those strategies to be translated into sophisticated laboratory protocols that require highly trained scientists to execute them under carefully controlled conditions, many times using sophisticated instrumentation to pull it off. What this work is demonstrating is intelligent agency is required to, in this case, for the origin of life to take place. And in fact, this is the theme of my book, Creating Life in the Lab. And I would submit to you that this argument that I'm presenting is a novel argument for design. Typically, when a Christian apologist would argue for design in a biological context, oftentimes it would be focusing on the inefficiency or the inability of evolutionary mechanisms to generate cells from chemical materials. Or it might involve looking at the features of biochemical systems and arguing that those features reflect the work of a mind. Nothing wrong with those two lines of argumentation. They're valid lines of reasoning. But what we're looking at with the argument from synthetic biology, if you will, is a different type of argument. It's an empirical argument where we are actually saying that we have empirical evidence that life requires the work of intelligent agency to originate. Because this is what's being demonstrated in the laboratory work that's being done, is that every study that's done in synthetic biology ultimately makes it very clear that the success is intimately linked to the work of that intelligent agent, the involvement of that intelligent agent. And so what I want to do this evening is extend the argument that I made in Creating Life in the Lab to not only the origin of life question, but to the history of life on Earth. And so to do that this evening, I'm going to talk about 4 studies that have been published recently in synthetic biology that basically make the same point that I made in Creating Life in the Lab. One of the studies is going to involve the origin of life. So this will be a way for me to recapitulate the argument that I made originally in Creating Life in the Lab, but focus on a more recent scientific study. And then we're going to look at a study in synthetic biology that gives us insight into the origin of new metabolic pathways in cells. We're going to talk about a study designed to understand the origin of eukaryotic cells. And then we're going to look at a study that gives us insight into the origin of the structure of the human genome. Now, I warn you that at this point, things are going to become a little bit technical. And I wrestled with how much technical detail to get into with this part of the presentation this evening. And for some of you, by the way, I'm not going to be technical enough. For others, I'm going to be just right. And for some of you, I'm going to be too technical. And I apologize, but I feel it's important to get into some of the technical details because the strength of the argument is actually in the details themselves. And so one time I was given advice by an editor when I was cutting my teeth as a writer, and that editor said, "Show, don't tell." And so what I'm going to do tonight is show you, not tell you. And again, that involves some technical details. So if at points in the presentation, if it gets just a little bit overwhelming, that's okay. Just take a mental break, and you can pick up when we move to the next module in the talk. But keep in mind that the big picture is, again, how much ingenuity is required to pull off this work in synthetic biology, okay? All right, so the study that I want to talk about first was published by a team of Japanese researchers. And these researchers created, in the laboratory, self-replicating protocells. Now, as a biochemist, when I read this paper, I just totally geeked out because this is really, really slick stuff. The ingenuity of this research project was, again, through the roof. This is science at its very best. What these researchers did is just amazing. And what they did is they developed these protocells that they could get to grow and replicate for multiple generations in a laboratory setting. In fact, they were able to couple the replication of DNA in the interior of the protocells to the cell or to the protocell growth and division. And then on top of that, they figured out a way to feed the protocells new nutrients so that it could support this process of growth and division and DNA replication for, again, multiple generations. So it's a really very slick experimental accomplishment. Now here is a cartoon that kind of illustrates what they did, and I'm not sure how easy that is to see, but what they did is they started with a protocell that is essentially a vesicle. So it's essentially a spherical structure that is hollow in the interior, where you have a vesicle wall, and then in the interior it's an aqueous environment, and that wall separates the aqueous environment of the interior from the exterior environment. So it's very much like a cell. And inside they encapsulated pieces of DNA, these nucleotides, which are the building blocks of DNA, and then also there is an enzyme they included called DNA polymerase that takes the building blocks and assembles them into DNA molecules. And to get the replication process started, they went through a thermal cycle where they heated up the protocells. This causes the DNA double helix to unwind. Biochemists call that a melting event. The DNA double helix unwinds, which exposes the single strand of DNA that then allows DNA polymerase to assemble from the nucleotides essentially daughter DNA molecules. Then they cooled the system down, allowing the single-stranded DNA to anneal into a double helix. So that's how they got the replication to work. They then added a compound that they designed and chemically synthesized called compound V, and they added it, and this compound V would become incorporated into the wall of the vesicle, and as a result of that, that would lead to the growth of the vesicle, and then it would cause the vesicle to grow to such a point it becomes unstable, and it fissures into two daughter cells. Okay? Then what they did is they had another type of vesicle that they made called a conveyor vesicle that is loaded with nucleotides that they would then add to the system. This would basically adsorb to the surface of the daughter vesicles that were produced, and then they would lower the pH of the solution, which would cause the vesicles to fuse, and voila, you're back where you started from. And they could, again, go through multiple generations before the system kind of faded. So really a pretty interesting, slick experimental design. Now, to pull this off, this is where you see the ingenuity come into play. Because in order to make the vesicles, to begin with, they had to very carefully select phospholipids, which were the components that would make up the vesicle walls. They very carefully had to select the phospholipids so that they had the right chemical and physical properties to form stable vesicles to begin with. They also had to develop essentially a synthetic route to design and synthesize this molecule V. And it turns out that this molecule V is a precursor that has to react with another molecule called C that they had to design, so that this would then break this into two components, one which would get incorporated into the cell wall, and another one which would be a leaving group that would thermodynamically drive the reaction forward. Okay? And then what they did, now this is a little bit more detail, is again they started with these vesicles that they created. They would go through the thermal cycle, and then you would wind up with the daughter vesicle. Now, because compound C is positively charged, they would actually add compound C at the time that they would form the vesicle. And because it was positively charged and DNA is negatively charged, you form these attractions that would then cause the DNA to become essentially intercalated into the membrane itself, into the wall of the vesicle. And this is how they coupled the DNA replication to the growth and division of the vesicle. They then added compound V, which would undergo a reaction catalyzed by compound C, and this would then become incorporated into the wall, leading to the division. And then with the conveyor vesicles, they had to add a phospholipid that had a negative charge, and again, C has a positive charge, so that causes the vesicles to essentially interact, and then with the pH change, they could get the vesicles to fuse. So in other words, they had to very carefully select phospholipids with the just right properties. They had to synthesize two non-natural lipids that had the just right properties that would allow essentially the vesicles to grow and divide and to couple the DNA replication to the growth and division. They had to very carefully adjust the composition of the vesicles to get the right amount of C in the vesicles, the compound C. They had to very carefully control the conditions for the encapsulation process. We didn't really talk about this. They had to very carefully regulate the heating and the cooling regime to get the DNA replication to take place. They had to have the just right amounts of lipids in the conveyor vesicles, and then they had to, again, very carefully regulate the fusion process. So I think what you can see from this description is how much ingenuity was involved in forming these protocells that could grow and replicate and couple DNA replication with the growth and replication of the vesicles. This involves, again, ingenuity. It's a very clever system that, again, displays foresight, displays this elaborate strategy that requires precise manipulation in the lab. And so by extension, when we think about the origin of life question, it makes more sense, I think, in light of this, to think that the emergence of life on Earth would have required the work of a mind. Because what this work is showing, again, is the central importance of intelligent agency. Okay, so now let's take a breath for a second. Are you guys tracking okay? Okay. Again, keep in mind the big picture. Keep in mind the big picture. Okay, now the next thing I want to talk about is the creation of novel metabolic routes. And one of the things that would have had to happen in the history of life from an evolutionary standpoint is that you would have had an organism called LUCA that would be the end product of the origin of life process. This would be the last universal common ancestor. But then for life to diversify from that point, that cell or that collection of cells would have had to evolve novel metabolic pathways, right? And what I mean by a metabolic pathway is that inside cells, there are these sequence of chemical reactions that convert starting materials to end products. And those chemical reactions are necessary to allow these cells to produce building block materials or other compounds that they would need to form cell structures, to carry out cell operations, or to break down materials, either to process wastes or extract energy from the environment. And typically, metabolic reactions involve a sequence of chemical interconversions where one compound is converted to another, converted to another, converted to another, and finally to the end product. And there are these proteins called enzymes that will mediate each of these chemical transformations. And so for a chemical metabolic pathway to emerge from scratch, you would have to evolve simultaneously a whole ensemble of brand new enzymes that would support, again, these different metabolic routes. But many evolutionary biologists actually think that a lot of metabolic pathways were jimmy-rigged together from pre-existing pathways, where portions of those pre-existing pathways were co-opted and then cobbled together to create new novel pathways. And so what we're going to do is look at how reasonable is it to think that evolutionary processes could do that by looking at a study done by researchers actually from UC Berkeley, where they genetically engineered yeast to make a compound called norscapine. Now, you may not be aware of this compound, but this is actually a compound used as a cough suppressant in many parts of the world. And the primary source of norscapine is opium poppies. And so this compound, again, is used as a cough suppressant, and people will harvest it from opium poppies. And you can imagine the regulations that are involved in growing opium poppies to produce this medicinal compound. But it turns out that recently, researchers have discovered that this compound has anti-cancer properties. And so people think that this could be a potential cancer drug. Now, this is really exciting because we have all kinds of safety data already available to us about norscapine. It's already used as a cough suppressant, which means that you can leapfrog a whole bunch of safety studies that you'd have to do before you could enter the compound into clinical trials. So people are really excited about this. And so this is a compound that is in clinical investigation now as an anti-cancer agent. The problem is that if norscapine actually becomes useful as a cancer drug, we've got a real problem. There's not enough opium poppies in the world to support the demands for norscapine. We just simply can't grow enough opium poppies to do that. Now, you can chemically synthesize the compound, but to scale up that process at an industrial level is so expensive that the compound's cost becomes prohibitive. So the idea is, could you take a yeast cell and engineer it so it has a metabolic capability of making this compound called norscapine? And so that's what these researchers did. And it turns out that, again, we understand a little bit about how opium poppies metabolically generate this compound. And so the research team from UC Berkeley took 10 enzymes from the poppies, or at least they took the genes from poppies, from opium poppies, that encoded the enzymes responsible for catalyzing the last 10 steps in the reaction that goes from a compound called scolarine to norscapine, and they incorporated those genes into the yeast genome. But what they wanted to do was make yeast able to generate this compound from glucose, from a simple sugar, and they discovered that in bacteria there is a metabolic pathway that will go from glucose to a compound called reticulene. So now all they had to do was go from reticulene to scolarine. And so what did they do? They discovered a metabolic pathway in rats that they could use to bridge those pathways. So they took a set of metabolic pathways from bacteria, rats, and opium poppies, and they incorporated those genes into the yeast genome and designed this metabolic route to create the compound that they were interested in. So this is a pretty elaborate metabolic pathway. But in order to pull this off, these researchers basically had to, again, they didn't create this from scratch. They had to, it required a detailed understanding of biochemistry. It required essentially foresight, right, and a measure of ingenuity to piece together this metabolic pathway. But then once they incorporated the genes into yeast, they discovered that they had to modify some of the genes so that those enzymes would work in the yeast environment. And then on top of that, they discovered that they had to regulate other metabolic pathways to support the efficient production of norscapine, because many times metabolic pathways can siphon off chemical intermediates, and if that happens, it shuts down the pathway of interest. So they had to go in and manipulate the regulation of other metabolic pathways very carefully to support the production of this compound, and then they had to play around with the growth media to be able to get the yeast to grow and produce high levels of the compound. So in other words, to generate a novel metabolic pathway, again, requires ingenuity. It requires knowledge, it requires foresight, and again, sophisticated manipulations in the laboratory. Now, the next study I want to talk about relates to the origin of what are called eukaryotic cells. There are two types of cells that exist in nature that are called prokaryotic and eukaryotic cells. So this is an example of a prokaryotic cell. This is would be what you would typify something like a bacterium. It consists of essentially a cell envelope, and the internal structure has no morphology. It is nothing but essentially molecules dissolved in the interior of the cell. These tend to be about 0.1 to about 10 microns in size. They're relatively small cells. The other type of cell that exists in nature is called a eukaryotic cell. And this is probably what you're familiar with if you pick up a biology textbook, because there's like a nucleus, there's all these internal membrane systems, there are these organelles. And so the big question in evolutionary biology is, how do you go from a prokaryotic cell to a eukaryotic cell? And the leading explanation today is something called symbiogenesis, or sometimes it's called the endosymbiont hypothesis. And the idea here is that you have a cell that would be a prokaryotic cell that over time would increase in cell size. And as it increases in cell size, it becomes, again, a massive cell that can engulf other smaller bacterial cells. And those smaller bacterial cells that become engulfed establish permanent residency inside the larger host cell. And they form a symbiotic relationship where the host cell provides nutrients for the endosymbiont, and the endosymbiont provides some nutrients for the host cell, and they form this permanent endosymbiotic relationship so that when that host cell grows and divides, those endosymbionts in the interior are also growing and dividing and get transmitted to the next generation of cells. And so evolutionary biologists think that after this kind of endosymbiotic relationship was established, over time the endosymbionts lost their genetic material and devolved to form organelles. So for example, mitochondria, which are these bean-shaped organelles inside the cell that are responsible for producing the compound called ATP. Should have turned off my phone and I didn't. The mitochondria produces a compound called ATP, which is a fuel molecule for the cell to use. And evolutionary biologists think that it was derived from a bacteria called alpha proteobacteria that, again, was incorporated into a cell, into a host cell, and then devolved to form the organelle, an organelle called a mitochondria. Now, one of the pieces of evidence that people will point to in favor of this is that we can see many examples of endosymbionts in biology, particularly among insects. Many insects have cells that have bacteria residing permanently within the insect cell. Again, these are endosymbionts, and so the argument goes, the fact that we can see those endosymbiotic relationships forming, it means that the first step in this evolutionary pathway, or this proposed evolutionary pathway, makes sense. And so recently a team of researchers wanted to understand what does it take to establish an endosymbiotic relationship. And so they did a study where they genetically engineered yeast so that they would be incapable of producing ATP, and they genetically engineered the bacteria E. coli so that it would be dependent upon the yeast's biochemistry to survive, and they essentially try to force these two organisms to form an endosymbiotic relationship. And so this is a cartoon showing what they did. This is a depiction of the yeast cell, and what they did is they basically disabled the mitochondria so that it would not produce very high levels of ATP. So now you've got this energy deficient yeast cell. Then what they did is they added E. coli into the cell, and the E. coli was no longer able to produce a compound called thiamine, but it could make use of the thiamine produced by the yeast cell. And then what they did is they genetically engineered the E. coli so that it would transport ATP from the interior of its cell to the exterior environment, establishing, again, this mutual interdependence between these two microorganisms. And so this is, again, forming this endosymbiotic relationship that people think was the first step in the origin of eukaryotic cells. Well, in order to pull this off, they discovered that it wasn't easy at all. It required, first of all, the researchers to identify what's called an ATP-ADP transporter, and the only place that you can find this kind of transporter that pumps ATP from the interior of the cell to the exterior environment are in endosymbionts. This kind of enzyme doesn't exist anywhere else in nature, and the reason it doesn't exist anywhere in nature is because it makes no sense for a cell to have an enzyme like that takes ATP from its interior and pumps it into the exterior environment. Why? Because ATP is the energy compound. If a cell is making this compound, it's going to want to retain it. It's not going to want to pump it into the environment. In fact, you'd expect those transporters to work in the opposite direction, where they would pump ATP from the environment into the interior of the cell. And so it almost looks as if endosymbionts are these highly unusual cells that have this just right property that allows them to form endosymbiotic relationships. It looks to me like they were designed to be endosymbionts, not evolved. But then what they discovered is once they did that, the E. coli would disappear from the yeast cell once they established the endosymbiotic relationship, and they realized that what was happening was that the lysosome, which is an organelle inside the yeast cell, was essentially engulfing the E. coli and digesting it and breaking it down. And so they had to block the lysosomal pathway. So what did they do? They went back to the endosymbionts and they found another set of proteins that block the lysosomal degradation pathway. So in other words, in order to establish this endosymbiotic relationship, again, it required knowledge, it required foresight, it required this elaborate strategy in order to pull this off. And again, the detailed manipulation. So even to form endosymbiotic relationships looks like it requires intelligent agency. It's not something that evolution is going to produce. Okay, one final study and then we'll bring everything to a close. And this has to do with the origin of the human genome. And this is a depiction of the human genome. And the human genome consists of 23 pairs of chromosomes, where one set of chromosomes comes from the mother, one from the father. And there are 22 autosomes, and then there are the two sex chromosomes, the X and the Y chromosome. And so this is what consists of the human genome. The human genome consists of 3.2 billion genetic letters. But they aren't formed in a continuous sequence of DNA, but rather are broken up into smaller DNA molecules that are complexed with proteins to form these structures called chromosomes. And it turns out that you can stain chromosomes, and when you do that, they form characteristic banding patterns like you see here. And this is an example of human chromosome 2. Well, it turns out that humans have, again, 23 pairs of chromosomes, but chimps have 24 pairs of chromosomes. And evolutionary biologists believe that chimps are our closest evolutionary ancestor. And the idea is that as humans and chimps diverge as separate lineages from that common ancestor, they believe that there was a fusion of two chimp chromosomes to form human chromosome 2. And so this is chimp chromosome 2A and chimp chromosome 2B. And when you align these chromosomes up, you see similar banding patterns, but also what you see in human chromosome 2 is near the center what are called telomeric sequences. The telomeres are regions at the very tip end of chromosomes, and they are designed to essentially stabilize the chromosome, to keep it from breaking down. And it's to keep chromosomes from fusing with each other. But it looks as if there's, again, this telomeric sequence in here that looks like it resulted from an end-on-end fusion. And then you also have in chromosomes an area called a centromere, which is where the mitotic apparatus binds and pulls the chromosomes apart during cell division. And you see two centromeres in human chromosome 2. One of them is active, one of them is inactive. And so on that basis, people argue that the human genome was shaped by evolutionary processes, one of which was the fusion of two chimp chromosomes to form this human chromosome 2. Now, in Who Was Adam?, I point out that I think the data does indicate what appears to be a fusion, but I raise questions as to whether or not that fusion actually was through unguided evolutionary processes. Instead, I suggest that that fusion is somehow giving us insight into what the Creator did, shaping our genome as human beings. And the reason why I say that is, in order for that kind of chromosome fusion to happen, it requires a sequence of highly improbable events. For example, as I mentioned, telomeres are designed to stabilize chromosomes. They're designed to keep chromosomes from breaking down. And they also are designed to keep chromosomes from fusing with each other. So if that's their purpose, this is what telomeres are doing, then it's highly unlikely that telomere-telomere fusion is going to happen. And if for some reason it did happen, as soon as you break, let's say you broke a chromosome at the very tip end, as soon as you lose that telomere, the chromosome becomes unstable. But if somehow you could get past that and get fusion to happen, you now have a mismatch in the number of chromosomes in that particular cell. And if this is happening in a sperm cell or an egg cell, the likelihood of producing a viable offspring is very low, let alone one that would be fertile. And so the likelihood of all these events happening is very remote. And so to me, it looks like there could have been a fusion event, but it seems to be something that, in my view, would be divinely orchestrated as part of a Creator shaping our genome as human beings, not something that evolution would produce. And there was a study published just recently by two independent teams of researchers who were looking at what does it take to fuse chromosomes end on end in yeast cells. And yeast have 16 chromosomes in their genome, and what they were trying to do is get these chromosomes to fuse to form a single massive yeast chromosome. And what they were doing is taking chromosomes and fusing pairs, so you'd go from 16 to 8, then from 8 to 4, 4 to 2, then 2 to 1. And one research team could get it to the point where there were 2 chromosomes. The other research team got it all the way to a single chromosome. But what they discovered is that you couldn't just randomly select chromosomes for this fusion process to happen. You had to actually very carefully select the chromosomes that you were going to pair up in order to get this to work. And the reason is because the chromosomes had to be chosen in such a way that the resulting centromere would be near the center of the chromosome. Otherwise, the yeast cell couldn't replicate. But on top of that, they discovered that they also had to disable one of those centromeres, because if there were 2 centromeres, then this would make it essentially impossible for cell division to happen. They immediately had to disable this, and they had to use essentially CRISPR gene editing to cut out one of the centromeres in order for this to work. And then they also discovered that they had to be very precise about cutting the telomeres off of the chromosomes as well, because if they didn't cut it in the right place, they would lose genetic capacity. They would damage a vital part of the yeast genome. So in other words, in order to get this fusion process to happen, it required a very precise manipulation of the chromosomes in order for this to happen, where you had to very carefully remove the telomere. You had to very intentionally remove one of the centromeres, and the right centromere, so that the resulting centromere would be near the middle. So in other words, if you had 2 chimp chromosomes fusing, the problem is now compounded by the fact that even if you did break away the telomeric sequences, it'd have to break away in the just right spot, and you'd immediately have to disable one of the centromeres as soon as that fusion happened in order for it to work. So in other words, in order to get chromosomes to fuse, it requires, again, intelligent agency. So in other words, what we have shown this evening is, looking at 4 studies involving synthetic biology, that not only does the origin of life require intelligent agency, but key transitions in the history of life would require the input of intelligent agency as well. And so this idea, again, that work in synthetic biology really allows us to make, again, a novel argument for God's existence. Again, and it's an empirical argument based on, again, looking at what does it take in the laboratory to affect these kinds of transitions that we think would have happened in an unguided manner through the history of life on Earth. What this work in synthetic biology is showing us is that it's anything but unguided, that the watchmaker isn't blind, but has to be fully sighted in order for these kind of transitions to happen.